Emergent Biosolutions is developing otlertuzumab, an anti-CD37 fusion protein for potential use in treating B-cell-mediated diseases such as chronic lymphocytic leukemia and non-Hodgkin's lymphoma. The structure of otlertuzumab comprises a single-chain protein therapeutic that has humanized immunoglobulin variable regions called scFE that are fused to a human IgG1 Fc region. Otlertuzumab is approximately 2/3 the size of a monoclonal antibody. B cells are a type of lymphocyte that originates in bone marrow and develops in lymph nodes. They can be distinguished from other lymphocytes, such as T cells and natural killer cells, by the presence of receptor proteins such as CD19, CD20, CD37, and CD52. These proteins are present on the surface of B cells at specific stages of B cell maturation and can be used to target B cell malignancies. CD37 is a cell surface protein from the tetraspanin family with 4 transmembrane domains and 2 extracellular loops. It mediates signal transduction, regulating cell development, activation, growth, and motility. CD37 is expressed on the surface of both normal and malignant B cells, including the majority of B cells involved in non-Hodgkin's lymphoma and chronic lymphocytic leukemia. This makes CD37 an attractive target for B cell diseases. Importantly, CD37 is absent or minimally expressed on T cells, natural killer cells, monocytes, and granulocytes. Otlertuzumab targets the CD37 signaling pathway involved in B-cell malignancies. In preclinical studies, otlertuzumab mediates B-cell destruction through 2 primary proposed mechanisms of action: antibody Fc-dependent cell-mediated cytotoxicity, or ADCC, and direct induction of apoptosis. ADCC is an extracellular immune process that causes cell death via an indirect mechanism. In ADCC, antibodies coat target cells, marking them for destruction by immune cells. In this process, otlertuzumab binds to CD37 on target B cells. Natural killer cell CD16 Fc receptors recognize the Fc of cell-bound otlertuzumab. Cross-linking CD16 activates the natural killer cell, mediating release of cytotoxic molecules such as perforin and proteases, known as granzymes, into the lytic synapse. Released in close proximity to a target B cell, perforin forms pores in the cell membrane. The pores create aqueous channels through which the granzymes can enter, causing B cell death. Otlertuzumab also causes direct apoptosis of normal and malignant B cells via a caspase-independent signal that appears to be distinct from that seen with anti-CD20 therapies. This pro-apoptotic signal is activated in B cells only when otlertuzumab binds to and cross-links CD37. In both in vitro assays and animal models, otlertuzumab demonstrates additive or synergistic killing activity of NHL and CLL cells with bendamustine, rituximab, BTK inhibitor, PI3K inhibitor, or rapamycin. After targeted tumors are eradicated, the body can be replenished with healthy B cells that are produced by stem cells in the bone marrow.